All right, let's do it. We're going to talk a little bit about developmental patterning. In this, we briefly hinted at like gradients and how, um, information of what a cell should turn into is determined by these gradients of these proteins or these morphogenic factors. Hox genes are very important in this regard, and we'll get into that a little bit more. But here's our MC Escher gastrulation picture, which kind of fits well because we have a lot of electromicrographs that are black and white, so it's kind of thematic, I guess. So we've been working on our gastrulation, and here is we're gonna look at Drosophila because a lot of these genetic studies have been done with Drosophila, just fast generation time, and a lot of this evo-devo means a lot of this stuff's conserved. It's not a vertebrate or amniote, but a lot of this very early stuff is kind of a model. In this case, the anterior of each gastrulation or gastrulating embryo is pointing upwards in the series of micrographs. Let me get my pointer. The first movement segregate, remember, the mesoderm, the ectoderm, and the endoderm. Here we see the ventral midline. This consists of about 1,000 cells, and it folds inward to produce this what we call a ventral furrow. So closing of that ventral furrow, the furrow is now pinched off to become a ventral tube, so something kind of like the neural tube. At this time, the embryo bends and starts to form the cephalic furrow, so cephalic being like the head, and this is on the anterior side. So then we also get this invagination that occurs, and it creates this pocket at the anterior and posterior of the embryo, and at the ends of the ventral furrow. So it kind of curves around up the backside. So we're looking at the anterior side right now. This is the dorsal view of the slightly older embryo, showing these pole cells down here at the bottom. They'll go on to form the midgut when they get invaginated here. So this is kind of a better view here. So we have this cephalic furrow, so the head's going to be up here. This posterior or future posterior furrow, future posterior right here. We have the furrow that goes around and would go up the backside there. So you can see this is the posterior, even though the anterior and the posterior are close together, it's curved around, right? So we have the head and then it curves up. So it's like kind of in a, you would think this would be the posterior, but no, because it's folded up like that, the posterior is folded into the front. So here we have kind of the areas of the fly. So we have the head, their mandible region, MX is the maxillary region, and so we have the head, we have the thorax, so T1, T2, T3, so the thorax regions 1, 2, and 3, the abdomen 1 and 2, and you can see it curves around, so abdomen 1, 2, 3, 4, 5, 6, 7, 8, so it's folded over on itself. Okay, so here's a little bit later in development. So again, we have the head regions here, and then we have the dorsal ridge, the thoracic regions, then we have the abdomen regions. And then as it unfolds, you can see, so this is kind of a newly hatched larva. Then we have the anterior segment here, so this is the head, and then we have the thorax and abdomen down that way. This is kind of a color view here of it. So this is kind of how it maps out to a fly once they fully metamorphosize, right? Because they actually do go through that because they hatch as a larva and then they turn into an adult fly. But we have anyways a head, then we have the prothorax, the mesothorax, the metathorax, and then the dorsal and the abdominal segments here. And you can see how they develop into each corresponding segment of the adult fly. So that's all good. A quick primer on fly development. But what's important is the idea of these patterns and this formation of these segments, right? So we have this maternal effect, this cytoplasmic polarity. So in this egg, we have a bicoid caudal gradient, but you can see that there is like very high concentration of this morphogenic factor on the left-hand side, but it dwindles down to nothing by the time you get to the middle. And then on the right-hand side, we have a very high concentration represented by this dark blue, and it starts to go down as well. We have another type of genes patterning called gap genes. So these form these big wide segments here. So you can see that we have like a very high concentration of yellow here, very high concentration of yellow here, but in the middle we have a very high concentration of a different, like a purple. We have parallel genes, and so these make these narrower bands. We also have segment polarity genes. So as you can see, at this point we have each of these like thoracic segments or abdominal segments here for the fly. You can see that it's always on this would be anterior, right? So this is the head. So it's wrapped around, so it looks kind of funny, but this is always on the head side of the organism, right? That they're expressed. So you could have the same thing, but with posterior too, and those would be segment polarity. So they tell you the anterior side of a segment or the posterior side of a segment. So let me make sure I covered everything here. So these proteins are transcription factors, right? So they are proteins that turn genes on. So it'll turn on the genes to develop wings or eyes or whatever that they're supposed to be in that section. So the kind of takeaway from this is that you have these sections that, based on their combination of all these different the maternal effect genes, the gap genes, the parallel genes, the segment polarity genes, that each section has its own kind of profile of what genes are turned on or off. And so that is what will allow differentiation of a specific cell to be developed in that spot, right? So this is just kind of a staining of anterior, posterior maternal effect genes. You can see it's very highly expressed in the anterior side and fades to nothing as you get towards the posterior side. So this is bicoid. We have these are gap genes. So this is hunchback, that's orange over here, and crouple, which is green. And areas where it's yellow is where both of them are. So you can see these form these kind of broader regions, these gap genes do. An example here is of, these are the parallel genes. This one is Fushitarazu, Fushitarazu, must be named after a Japanese scientist, I would assume. Anyways, so you can see they're expressed in these narrow bands that helps you get even more detailed of what your location or your profile is. And so this is segment polarity here. These are engrailed is the name of this one. You can see they form these bands right on the edge of each of those forming segments. And then lastly, I have kind of a cheat sheet for what these genes are, what they do. So maternal effect genes are these mRNA that localize to different regions of the egg. The gap genes are the first ones expressed, and they cause those big bands. Parallel genes then are next expressed, and they divide the embryo into periodic units, so like smaller stripes of 7 bands. Segment polarity cuts everything at the front or back of each of the each unit or each section of those developing sections of the fly. And these homeotic selector genes are genes who determine the developmental fate of each segment. So that's just like a general term for this. So that's all. I just want to give you a brief. We'll go a little bit more into these developmental patterns or whatever, but these gradients, how you get specific tissue types, and are very much based on these local gradients. And we know a lot with the fly, so we're versus humans. So we're going to do a bit more of this with fly as our model to kind of get an idea how this all works. So thanks for watching, and I'll catch you in the next video.